Hi, I'm Barbara Osborne from the University of Massachusetts, and this is my collaborator and my co-founder, Catherine Knight from Loyola University in Chicago. We want to take a minute to thank both Equalize and our mentor, Anil Goyal, for all the help they've provided us over the past several months. We just wouldn't be here today without their help and advice and holding our hand. So today, what I'd like to do is to introduce you to Hasentech and our technology. Recently, Hasentech has discovered a way to dampen inflammation in a variety of multiple diseases, some of which are shown here. We have decided, however, to focus on one particular disease, and that is graft versus host disease. What Hasentech has discovered is a compound that's isolated from commensal bacteria that blocks inflammation in these multiple diseases. So why are we focusing on graft versus host disease? It turns out, as many as you probably know, bone marrow transplantations are used to treat blood cell cancers. And these transplants are actually curative in many instances. However, graft versus host disease, or GVHD, is a major complication that occurs with many bone marrow transplants. Indeed, of the 50,000 people worldwide that receive bone marrow transplantation, about 40% of these people actually develop GVHD, and 50% of those people die from the complications of the disease. So there's a high risk in having a bone marrow transplant. Turns out this is really quite personal to us because we lost a colleague following transplantation and treatment for lymphoma a few years ago. And many of you probably know others who have suffered from this disease. It's really rather devastating. So what can we do about it? Let me tell you a little bit about the disease first. So what is done during bone marrow transplantation is a donor is first identified. And from that donor, bone marrow is isolated. It turns out that bone marrow is rich in hematopoietic stem cells, and that the graft from the bone marrow is injected into the host, where the tissue, the stem cells, develop into new blood cells and repopulate the host tissue. It also turns out that within this graft, there are immune cells which can recognize the host as foreign and attack the host and mount the disease we call GVHD. The organs that are attacked most frequently are the skin, the bowel, and the liver. So currently what happens before transplantation, patients receive toxic immunosuppressive drugs just at the time of transplantation, and sometimes frequently a couple of doses later following the transfer. Hasentech has a different approach. We want to drug the cells, not the patient. We don't want to systemically immunosuppress the patient. Let me tell you a little bit more about our solution. I told you that Hasentech has developed an anti-inflammatory drug. This is called HTO1. It's a natural exopolysaccharide that's been purified from the bacterium B. subtilis. The Knight Lab has shown in a variety of different inflammatory situations that HTO1 blocks inflammation. And they recently have derived a structure or determined a structure for HTO1. What Hasentech would like to do with some funding, if we are lucky enough to receive some funding, what Hasentech would like to do is to chemically synthesize this molecule, develop a new compound, a chemically defined compound we'll call HTO2. I should mention HTO1 is covered by a UMass patent that was recently issued. And we would suspect, and we would hope and predict that HTO2 would form a new novel compound and thus new patent. For me to tell you about the mechanism of action of HTO1, I first have to take a moment and tell you about how T cells get activated. So what happens when T cells get activated is first an antigen presenting cell, here represented as a dendritic cell, presents antigen to a naive or resting T cell. That T cell quickly becomes activated. It starts to proliferate and divide and secretes inflammatory cytokines. And in the case of a GVHD, it's this inflammatory response that drives the GVHD. What Hasentek has found is if we treat dendritic cells with HTO1, these dendritic cells no longer activate T cells, but instead block T cell activation. So what this has led us to predict is HTO1 treated dendritic cells will block T cell activation and prevent GVHD. And because inflammatory T cells are the real culprit in GVHD, we feel this approach could really help us in blocking the deleterious effects of graft versus host disease. To test this, we first started with a mouse model of bone marrow transplantation. And let me point out, in this experiment I'm showing you here, the animals were treated with HTO1 at the time of transplant, and they were treated IV. So they received an IV dose of HTO1. And as you can see, the two control groups of animals died quite readily and very quickly, whereas the HTO1 treated animals, many of them survived out to 80 days. In fact, there was 70% survival versus a 20% survival in these studies. These data pushed us to go and look at this in a murine model of human GVHD. So in these experiments, in our humanized model, what we did was we produced dendritic cells from cord blood cells that we can obtain commercially. We produced dendritic cells and then treated them with HTO1 overnight in a tissue culture dish. They were then mixed with the human donor cells and injected into our humanized mouse model. And as you can see here over on the right, when you look at the data, again, the animals in the control groups in red and blue died pretty readily, whereas the animals that received HTO1 dendritic cells survived, many of them survived out to 60 days. I want to point out two things here that are important. First of all, this is HTO1 dendritic cells delivered with the graft. HTO1 is not given to the animal, just to the dendritic cells. And the other thing I want to point out here is this is one dose of HTO1 dendritic cells given at the time of transplant. We now would like to seek funding to test the possibility that giving a second or a third dose may even enhance the survival and provide more protection. So we feel that the Hasentek innovation is pretty obvious. Current therapy right now treats patients with multiple immunosuppressive agents. And these patients, as I said, they get a large dose of an immunosuppressive cocktail that remains with them for several months following the transplantation. Hasentek, in contrast, treats these dendritic cells ex vivo with HTO1 and avoids systemic immunosuppression. So we're treating the cells and not the patient. We see that there are two major advantages for Hasentek. The one that I just mentioned, no systemic immunosuppression of the patient. But the other major advantage is we can have an off-the-shelf product, HTO1 treated dendritic cells that can be provided to transplant centers to block GVHD. Additional advantages we feel are that HTO1 is derived from a harmless probiotic bacterium, and HTO1 suppresses many other inflammatory diseases that I mentioned briefly at the beginning of my presentation. We've received funding from NIH in an STTR grant. We've also received funding from UMass Manning IELTS funding, and we have two other grants, one another STTR, as well as another Manning IELTS grant pending. We feel that the market opportunities for Hasentek technology are pretty obvious. This is a disease that kills a lot of people. People choose not to undergo bone marrow transplantation because of the problem with GVHD. If we could solve the GVHD problem, we could go from 50,000 bone marrow procedures a year up to 2,000 bone marrow procedures a year, we estimate. And we did a little bit of back of the envelope calculation that if the Hasentech technology costs $10,000 per patient, this could be an annual market of over $50 million per year. Our development plan is multifaceted. We want to fine-tune the timing and dosage of HTO1 treated DCs. We want to contract the synthesis of HTO2. And in particular, we want to raise funds to hire a scientific and management team and develop a business and regulatory approach. We'd also like to continue our ongoing studies to define the mechanism of action of HTO1 and HTO2. You've met Catherine and myself, two founders of Hasentech, but the third founder is Richard Goldsby, shown here. Dick and Catherine and I have had a long and fruitful collaboration over several decades. Most notably, Dick and I created a company with two colleagues in 1999 called Hematech. Hematech was sold in 2005 to Curin Pharmaceuticals. And Catherine was a member of our scientific advisory board. So we've worked together over 2 or 3 decades in translational immunology. Dick is also the chair of our scientific advisory board, which includes Dr. Patrick Stiff and Dr. Lisa Minter, who have expertise in bone marrow transplantation, as well as animal models of graft versus host disease, respectively. So I'd like to conclude by saying we feel strongly that HTO1 and HTO2 represents a novel approach over the current GVHD treatments. We have assembled a team that has worked together for many decades. It's experienced in the field of immunology, and we are committed to the translation of HTO1 and O2 to the clinic. We feel that an infusion of approximately $600,000 would allow Hasentech to de-risk the technology and attract major investments. Additionally, we hope that this infusion of money would allow us to get to the point where we can turn one of our phase 1 STTRs into a phase 2. In conclusion, we feel it's likely that Hasentech has the technology to treat a number of inflammatory diseases, including the GVHD that I just mentioned, as well as IBD and Crohn's, asthma, and autoimmune diseases. And with that, I'm willing to take some questions.
Thanks so much, Barbara. Chris Borner.
First of all, a very nice presentation. GVHD continues to be an area of significant unmet need. And interestingly enough, there was recently an approval of abatacept in GVHD. And as you may well know, abatacept inhibits T-cell activation by binding to CD80 and CD86. So I think you've got a little bit of proof of concept there. Can you talk a little bit more about how you would think about the development of this asset? And then any ideas beyond GVHD? You just briefly mentioned it there, but maybe could you elucidate on that just a bit?
Sure. We've gone back and forth, and in fact, it was the Equalize program that made us focus on GVHD, because we came to the conclusion that this was the greatest unmet need. And the fact that we can use the HTO1 ex vivo and not give it to the patient really pushed us towards focusing on GVHD as our first product and first attempt. We have looked, Catherine's lab and my lab have looked at animal models of multiple sclerosis, EAE. We have looked at, Catherine, I haven't, but Catherine in collaboration with another colleague has looked at asthma. And we both have looked at IBD and Crohn's. But in all of those instances, maybe asthma, you could actually, in fact, I know with asthma, you could give it nasally. But the other diseases that I mentioned, it's going to be an injection of HTO1 or HTO2. And we decided that the ex vivo approach with GVHD made the most amount of sense as our starting product.
Thank you. Very helpful.
But we sincerely believe there are anything that has a serious inflammatory response, we think could be treated with HTO1.
Yeah, nice presentation.
Thanks.
Mr. Abidin.
Sure. Yeah. Chris kind of stole my question, but yeah, let me just elaborate more on the broader application. I think when you think about investors, they're also thinking about where specific programs might fit for other strategic acquirers or collaborators, et cetera. Mm-hmm. And GVHD, I think it's great that you highlighted that you have a clear plan, great preclinical work. I think bringing forward, because I didn't know if you had these other data in these other IBD, asthma, et cetera. And if you're showing concordance of good activity across that, I think that makes a more compelling presentation. And also you may have investors who want you to do a little more work to advance those applications while you pursue the lead, because you have kind of a pipeline in a product, if you will, if this works. And also shots on goal, these are tough diseases to crack. And so sometimes in a case like this, you do want to pursue more than one indication. I'm just curious, the 600,000 to de-risk the program and make it more attractive to bring larger, that seems light to be able to do the work, to be able to raise more money and make it more compelling for a larger round. And it may be best interested for you to not dilute yourself, too much. And so just talk to me about what you'll do with the 600,000. What would you do if you had 2 million, right? If you could.
Yeah, sure. I mean, we could do a lot more with 2 million than we could with 6,000, obviously. Now I'm going to say something that maybe isn't so politically correct, but this is the problem that girls have. We sometimes don't ask for enough, and I've tried to learn better about that. But we did really seriously, Catherine and I seriously sat down and asked, what would we need to accomplish what I put on our development slide? We feel that that would give us 2 years of funding. It would allow us to hire 2 individuals. We have 2 individuals working in our labs that we have identified who would be excellent business and scientific partners for us in the company. We could pay their salaries, and we could do the experiments that we've outlined in that development plan. We could do a whole lot more. I mean, that would all be within GVHD. We could do a whole lot more with more money. We actually went back and forth. Should we include a variety of different inflammatory diseases? But with 10 minutes and having to explain each one, we decided to focus on one. And for lots of reasons that I've already articulated, it was GVHD.
Yeah,
you're absolutely right. We'd like to have a lot.
Ask for more. That would be my advice. Ask for more. Don't be bashful.
Thank you.
Yeah.
Megan.
Yeah, thanks. Apologies if this was addressed a little bit already, but just as clarification, the approach with ex vivo treatment of the dendritic cells versus actually injecting HTO1 or 2 into a patient. Sorry, can you remind me the rationale behind that again?
Sure, because whenever you don't have to put a drug in a patient seems to be a good approach. And if you can get your effect by treating the dendritic cells. So what we're really doing is we're treating dendritic cells that are in essence tolerogenic, and they are blocking the in vivo responses within the host. Now, we don't know, and these are some of the experiments we have to do in the next year or so. We don't know how long those dendritic cells will block the GVH. We assume that we could probably dose it so that it's longer or give multiple doses of it. And those are the kinds of questions we want to ask. But we feel that creating a product that does not have to be delivered to a patient, but rather to cells that are then given to the patient is probably a lot less risky from an FDA and approval point of view.